So it's time for our last speaker of the day, Jean-François Aubry. So he's a research director at CNRS in Paris and has a PhD in physics. He's interested in ultrasound imaging of the brain, medical application of ultrasound, and ultrasound-based therapy for brain. And so please welcome Jean-François. You want to connect your computer?
Oh, I need a laptop. But I can. There was another laptop.
It's here.
I think I connected it. This is not good?
No. Oh, yeah. Okay. That's good.
Might need to switch.
You should do it outward.
With the Macintosh.
No, also with this. Especially
Yeah, I know. Oh, there you
Yes. There we are.
go. Sorry, we took some of
Yes,
it.
maybe. Oh, and now I. Thank you very much for the introduction and for having me here in this great symposium. I will start with a disclosure, well, a couple of disclosures actually. I currently have an ongoing research collaboration agreement with Insightec, which is the world leader on MR-guided therapy, and our agreement is on MR-guided brain therapy. And I'm also a consultant and shareholder of First Mobile in the US, treating low back pain with ultrasound. So, starting with transcranial ultrasound therapy in general, there are actually many ways to use ultrasound for brain therapy. So basically brain surgery, blood-brain barrier opening, and neuromodulation. All of them can be done with ultrasound. The first one is thermal therapy, where we would use actually continuous sonications over several seconds to increase the temperature of tissues and coagulate tissues. It can be done through the skull, deep in the brain, and you can have a millimetric precision. So we've been involved a lot in this field, but you will find lots of literature, mainly from a group in the US at Brigham and Women's Hospital, and it has been successfully translated into the clinics for the past decade. You can also use pulsed sonication in conjunction with injection of microbubbles that are used as contrast agents for diagnostic ultrasound. So it is approved for diagnostic ultrasound, but when you inject those bubbles, ultrasound pulses will have the bubbles oscillate, and you can stretch actually the vessel walls in the brain, and you see here that the contrast agent goes into the muscles here on these MR images, but does not penetrate the brain, except at the location where we focused ultrasound with those pulsed sonications. So again, we've been involved in this field for the past 20 years, but the seminal work was done by the group of Kullervo-Hininen in Boston 20 years ago. And you will find also many works, and now this has been translated also into the clinics. Last but not least, if you use pulse sonication without microbubbles, you might also induce ultrasound neurostimulation. And we've been involved in this field for almost a decade now, working with rodents first and then translating to non-human primates, and I'm going to focus on this in this talk. And of course, again, many other teams also worked on ultrasound neurostimulation. Now, to give you an overview of what we do in Paris in our group. So we work on those 3 applications, and we actually apply it to rodents, non-human primates, and humans. And I'm going to explain to you why we need actually to adapt to the size of the head of the animals and the thickness of the skull. So we developed transducers dedicated either for rodents or non-human primates or for humans. And we have all those available in Paris. So ultrasound neurostimulation actually is not new at all. It was introduced in the 50s by Frank and William Fry, the Fry brothers, where they were able to introduce reversible changes in the central nervous system with ultrasound in cats. So it's not new, and this was the proof of concept. But unfortunately, actually, if you look here, what they claim is that the intervening skull had to be removed. They could not do it transcranially. The reason is that when you do not have any skull in place, if you use a focused transducer, it will concentrate the waves nicely into the medium, as is done actually for obstetric imaging, for example. But as soon as you have a skull in place, because of the difference in the speed of sound in the skull and the thickness of the skull is varying, the wavefront will be distorted when focusing through the skull. And so it means that you will have sometimes no focus, 1 or 2 foci, and it's going to be shifted away due to the refraction of the skull. So the first challenge here is to actually be able to focus ultrasound waves precisely through the human skull. And what has been proposed in the past is to use arrays of elements composed of typically hundreds of elements. The InsightTech machine is using more than 1,000 elements and adjusting actually the delays so that we can compensate from the effect of the skull in order to refocus. Now, how to learn how to compensate those effects. So first, we need to somehow estimate the effect of the skull. And we've been working actually together with Hininen's group, so for almost 20 years now, working on ways to predict how the skull is going to affect the ultrasound. And the idea back there was to use CT scans, and based on those CT scans and based on the Hunsfield unit, be able to deduce the acoustical properties of the skull, speed of sound, density, absorption, so that we could feed the wave equation, and we can simulate the propagation of the wavefront through the skull. When we do so, if we forecast the effect of the skull, we can compensate for it. And you see here in this illustration, this is without any correction. So you see, of course, maximum pressure is around focus, but you see that by adjusting the delays, you have a much tighter focus, and you focus ultrasound exactly at the location where it was targeted. But look also at the pressure amplitude here on the scalp. You see that here the ratio between the scalp and the target is much better when adjusting the face. Most of the work has been done based on CT images of the skull, but you know that it's possible also to use MR images. So we made a proof of concept a couple of years ago at the University of Virginia. You see here a skull. This is a photograph, and this is the MR image that we got from the same skull. And we used those information from the MR image to feed the InsightTech machine. And you see that the correction that was calculated by the machine with either the MR-based images or the CT-based images are similar. And actually, when looking at the efficacy of the thermal rise at focus, we see that CT and MR-based correction is pretty similar. But currently, the InsightTech machine is only approved for CT images, but we know it works also with MR. So this is for I would say, humans. The good news, though, is that for rodents, the skull is thin enough that it does not affect the wavefront so much as compared to humans. So lots of experiments have been conducted on rodents on neurostimulation in the past. And you see here the effect of the frequency. So if you increase the frequency, you increase the resolution. And you see that overall, the effect of the skull is not so important, except for those stripes here. And this is because of the standing waves that we induce in the head of the animal. And I insist on that again. The skull is thin. So the problem is not really the skull here, but more the head, because the wave is completely reflected at the bottom of the animal's head. So still, it is possible to focus ultrasound, even at rather high frequencies on rodents. And this is the reason why many teams have been studying transcranial ultrasound stimulation on rodents with lots of different transducers, different frequencies, and being able to show the proof of concept of neurostimulation. Again, you see that the wavefront here inside the brain can be quite complex, but still, we were very surprised to see that we could induce very specific effects on animals. You see here, when ultrasound is on, and only when ultrasound is on, you see a movement of the tail of the animal. If we increase the voltage, so the pressure, you see that we increase also the effect. So the movement of the tail was more important here. Now, if we change the location of the ultrasound beam, you see now that the eyeball here is moving and the tail is not moving. You see the animal is not moving at all. We induce only a slight movement of the eye towards the nose. And third example here is the whiskers, a couple of whiskers moving only when ultrasound is on. And so this was, to us, the proof of concept. We wanted to duplicate the results from Will Tyer group in the US. And the only problem here is that we have a very limited time window to do those experiments because the rats are anesthetized. And of course, if the rat is deeply anesthetized, it's not going to move, whatever you do. So we can only perform those experiments just before wake up. So typically during 15 minutes twice a day, which is not enough to explore all the parameters. So it's the reason why almost immediately we switched to awake animals, and we worked on monkeys. And on monkeys, we decided to target the frontal eye field. Again, so the skull bone is in between rats and humans, but still, you can see here that for the frontal eye field, the simulations show that we can actually focus the beam quite precisely with ultrasound. And the reason why we chose this target is because we wanted to study voluntary eye movement. So basically, the monkey was trained to look at the screen. When a target would appear on one side of the screen, it would be trained to look at the opposite direction. And we measured the latency time. So basically, if the monkey were looking at the target, it would be actually a reflex movement. And what we want to do is to have the monkey process it and look at the opposite direction. It's called an anti-saccade. The control of voluntary eye movement is complex. It involves the frontal eye field, the supplementary eye field, also the ACC, so many areas in the brain. We decided to target the frontal eye field and see how we might change the latency time. So here is the setup, and this is how it was done. First, we would start with baselines to look at the latency time of the animal for one given day. And then, again, at the end of the experiments, we would also do it 100 times again to see if the baseline is the same. And in between, semi-randomly, sometimes we would sonicate with ultrasound 100 milliseconds after the target is appearing on the screen. And then we would measure the time it takes for the animal to look at the opposite direction. So typically you see, of course, the transducer was placed on one side of the brain. So when the voluntary eye movement was on the same side as the transducer, it would be called ipsilateral movement, and contralateral if the target was appearing on the other side. And actually what was expected was to see a change in the latency time for ipsilateral targets, but not for contralateral targets, which was confirmed by the measurements. So this was our initial paper on primates, and we wanted to have a closer look to the effect of ultrasound in the brain and the neuromodulation effects. So same task, same monkeys, but we added also microelectrode recording sites in the supplementary eye field. We didn't want to measure the activity at the location where we were sending ultrasound because we expect micrometric movements of the tissues because of radiation pressure. So we didn't want to induce artifacts with ultrasound. So we are stimulating the frontal eye field, recording the supplementary eye field. And here is an example of the typical recordings that we saw. Blue is before ultrasound. So here is, so we had some firings, of course, in time, but each time we perform the anti-saccade, we saw that this neuron here that we are recording actually was not involved. It was not recruited when doing the anti-saccade, but when ultrasound was on, it was actually firing more. So we have an increase of activity here of a neuron that was less active during an anti-saccade. And we saw also the opposite. So if we record the activity on a neuron here in blue that is activated during the saccade, we see a decrease in activity. It's not completely shut down, but the activity is decreased during ultrasound. And on average, we see actually changes in activity when ultrasound are on and when we are sonicating into the frontal eye field, but when we are sonicating in a control area that was a motor movement but not involved in the visual task, we see that there is the firing rate remains unchanged. So this was, I would say, immediate effect of ultrasound. When we were doing those experiments, every 2 seconds or so, we might have ultrasound ongoing, and we saw that there was no impact from one sonication to the other one. So immediately after ultrasound, we don't see any change in activity. And we all want to induce long-lasting effects in order to be able one day to use this technology to treat patients. So we changed, and instead of using very short impulses with ultrasound, we proposed to use bursts of ultrasound, so pulse stimulation lasting for 20 seconds, and then we would stop. And in that case, again, same anti-saccade task, and we see the same. For ipsilateral movement, we see a significant change in the latency time, and we don't see such change in contralateral direction. The big difference, though, is that a 20-second ultrasound were applied here, and the effect is lasting for 20 minutes. So it's different. You see, we sonicate once, and we induce a long-lasting effect. So the effect was typically lasting for 20 minutes. And this is when we, at the beginning of those experiments, we had some discussions with the Oxford group from Matthew Rushworth, and namely with Lennart Verhagen as well. And they wanted to use ultrasound and look at changes in connectivity with MR, functional MRI. So in that case, we knew that we would have to induce an effect that was lasting at least for an hour. And it was good because when we started those experiments, you see here that at the end, of the experiment, we go back to the resting state, I would say. And we wanted to see it. We did not want to see an effect and not go back to baseline. So we started with 40 seconds, and we saw 40 seconds sonications. The effect was lasting too long, and the monkey would stop the task because it was too tired, and we would not go back to normal, to baseline. So we went back to 20 seconds to see the return to baseline. So together with Oxford, we used the 40 seconds sonications. And here we showed changes in connectivity on the brain that lasted for at least more than an hour, or almost up to 2 hours. And basically, if you look here, if you do a seed-based analysis, the seed is in the SMA, and we would here, for example, apply ultrasound stimulation either on the SMA or the FPC, we have completely different results. For frontopolar cortex stimulation, we don't see much difference between the two. Again, the seed is in the SMA, but when the seed is in the SMA and we stimulate the SMA is where we see connectivity changes. And we did the opposite. So if the seed this time is in the frontopolar cortex, you see that when the ultrasound is applied to the SMA, there's little change in connectivity, but there's more change in connectivity when ultrasound is applied where the seed is located. In collaboration with Matthew Rushworth's group, they did also additional experiments targeting the amygdala or the anterior cingulate cortex. And we look at the extent. So where do we see mostly changes in connectivity? And you see that, again, this is around the amygdala when we sonicate around the ACC when we sonicate in the ACC, and around the amygdala when we sonicate in the amygdala. The thing is, if you have a closer look and you look at the big picture here, you see that the focusing is quite good on the outer part of the brain here, on the cortex for the frontal eye field experiments. It's okay for the SMA, but you see here that actually we see stripe, we see standing waves, we see blurring of the focusing in that case. And this is due to the fact that even though the monkey skull is thinner than human skulls, when trying to focus deep in the brain, it gets more difficult. And so the fact that it's easier to concentrate the beam on the outer part of the brain is the reason why the first in man neuromodulation occurred in the cortex. It's not optimum because what is most interesting is that focused ultrasound should enable you to modulate activity deep in the brain non-invasively, but it was just easier to achieve. And this is an interesting study from Sung-Chin Kyo group, where they stimulated the somatosensory cortex, and they were able to induce sensations in the hand of the patients. Those were various sensations. It depends here on the volunteer. Most of them had tactile sensations, except for one. And by looking at the thickness of the skull, this is the one with the thicker skull. So it might be the reason why ultrasound dose was lower for that patient, that volunteer, and they did not see an effect. But you see that they were able to induce sensations in most of them. Similarly, Tyler group also, so this experiment is more complex. They stimulated the median nerve, look at the response of the patient, the volunteers, recorded the EEG, and they compared the EEG response to nerve stimulation when they were at the same time stimulating the brain or not. And you see some part here of the EEG in gray is where the result was statistically significantly different between sham and TFUS by sonicating here the somatosensory cortex. Since then, there's been more than 20 studies on volunteers with various applications, various targets. To be honest, I mean, whether this or the results here are kind of disappointing compared to the promises of ultrasound stimulation. Something we need to keep in mind, so some of them were cortical stimulation, others were deep stimulation. And if we look at a few of them here, we see that the focusing was not as good as one could think. This is an example. So they were supposed to have only one focus. So we see that the focus is steered and it's split into two. Same thing with Brinker, Badran. And actually, if you look in the transverse plane, again, you see that the difference, we did not show the difference between the targeted point in red and the maximum of pressure amplitude can be off significantly. Even though this one is cortical, this one is deeper in the brain. So the defocusing effect of the skull cannot be neglected. So I would say it's a nice try, but like that, I think with a single element transducer, it's not precise enough to make sure that you stimulate the right location. Again, we know that it's possible to compensate for all those the stimulations. So why not give it a try with those devices, which is what we did. We had actually 15 patients scheduled for essential tremor treatment with thermal ablation, so with the InsideTech machine. So you know this is the tremor of the patient before the sonication. So we install the system. Patient is on the table, and what we do is that we create a lesion in the thalamus, in the VIM, with ultrasound, and you see that after creating the lesion, the tremor is much lower, and you can see here this is before we asked the patient to write the date, the treatment date, and you see that after the handwriting is much better. It's even gaining confidence. He realizes that he can write quite nicely, and the year is written very nicely. And what we did is that on top of this experiment, we assessed the tremor during treatment by adding accelerometers on the patient hand inside the MR. So those are MR-compatible accelerometers. And what we wanted to see is quantify the tremor inside the MR. So during treatment, again, so this is not neuromodulation yet, this is thermal ablation. Thermal ablation, you see this is the first sonication with quite a high power. So the maximum temperature was 51 degrees in the brain. And you see a decrease between the beginning of the sonication and the end of the sonication. You see a decrease in tremor as we measured with accelerometers. But 51 degrees is enough to induce an effect that is not permanent. So a couple of minutes later, when we try again by increasing the power, we see that the tremor was back, actually. But this time we reached 59 degrees, and you see again a strong decrease in the tremor, and you see that now the tremor is gone. So here was, I would say, a transient effect. And now this is the permanent effect, and the treatment was, the patient was treated. So on average, we had more than 90% reduction, which hold in time for many months. But the acoustic energy that you need to achieve this lesion is a lot. It's 20,000 joules. And here is the planning on the patient. So this is the target. And you see here is how we do it. First, we align, we make sure that we sonicate at the right location with very low power, and we verify, and then we increase the power. What we did is and that's the reason why we added those accelerometers, is that during the alignment phase, right after the alignment phase, once we know where we are, we tried to use the low frequency, low intensity ultrasound. So pulsed sonication, we could not measure any thermal rise, but we wanted to see if we were able to decrease the tremor. As I showed you before, when you look at the changes in EEG recording that was induced on humans, changes can be seen, but they are very low. So I would say if we would induce a 20% reduction in tremor, it would be a good result. And what we saw, actually, we were surprised to see that actually what we induced is a 98.2 decrease in tremor with 8 joules, so almost no energy, very low power, extremely low power for a few seconds, and the effect lasted for 30 minutes. And actually, for this very first patient, the patient and the neurologist who was inside the MR thought that the treatment was actually done. That we switched to high power, burned created a lesion, and that the treatment was done and the patient would go back. And actually, that was not the case. So this was mind-blowing to us. It was way more than what we were expecting. And the thing is, we did it on a second patient here. And you see, once again, this is the baseline. And this is right after the neuromodulation sequence. So again, this is by operating the Insightec machine at its minimum level. And as we knew this one, that this is what we might expect, so we measured several times, almost every 5 minutes, the tremor. And you see that the tremor was significantly reduced for half an hour. And as soon, of course, the neurosurgeon got anxious because she wanted to treat the patient with high-power ultrasound. So as soon as the tremor was just back, just enough to be able to have some feedback from the patient, we did the treatment. But again, so this was also a 97% reduction for 30 minutes. We could not wait more because we had to treat the patient. So I don't know how long the effect would last, but at least it was less than 98%, and then we could treat. But if you compare to the 90% reduction, it's a lot, and this is for the final treatment. So, of course, for the patients, what was important was this, because then they've been treated, they went back home, they had no tremor anymore. But what was important to us was this proof of concept, that if you know where you are focusing ultrasound, you can induce a very significant effect and have a strong target engagement. I was surprised to see last month at the Focused Ultrasound Foundation, where we presented those results, another group, and I didn't know before, was also using the Exablate neurosystem at low power to stimulate the nucleus accumbens to treat patients and to reduce craving. And what they did is that they showed them pictures of either opioids or cocaine or alcohol and look at the response of the patient, and they quantified craving. You see here is sham sonication. So this is alcohol. One of them is opioid, and the other one is cocaine. Here is the effect of the sham sonication. And you see here now, 5 minutes after sonicating in the left nucleus accumbens, you see a strong decrease of craving, almost down to no craving at all. And then they also stimulated the right nucleus accumbens. This is for the first patient. Second patient, you see the same. Craving, went down to 0. So again, this is a very strong target engagement for a completely different pathology. And So again, this is great to show now that there is even stronger expectations on the translation of ultrasound neurostimulation to humans. Now, what is the price to pay? Well, it's a lot. Using this machine, we bought one because we wanted to use it for thermal ablation. So of course, you need a lot of power, and you need to make sure of what you're doing, but it's typically 1.5 million euros to treat the brain. Compared to other neurostimulation techniques, it's way too expensive. The thing is, so here is when you use a single element transducer, and the tendency for the past 20 years has been to increase the number of transducers to achieve a more and more precise focusing. And 7 years ago, we just thought, okay, what about using one single element transducer, but no compromise on the efficacy of the correction through the skull? If we could, with one transducer, create the exact shape of the ultrasound beam as the one we create with multiple elements, then we would be able to compensate the skull aberrations. Physically, it's impossible like that, but if you apply an acoustic lens in front of the transducer, and if you calculate the thickness of the lens that would create the same wavefront, it should work. This is what we patented 7 years ago. And actually, yeah, it works. We showed on dried skulls that by using this lens, again, this was on a single element transducer, so we have no choice. No lens, no correction. So no correction is what we have here, but with the lens, we see that we are able to correct the aberrations induced by the skulls. And on average, on 3 different skulls, what we see is that the maximum intensity at the target is 10 times higher with the lens as compared to without the lens. And the potential is huge. Remember the defocusing that we were seeing before on those studies. This is without a lens, and this is with the lens. Again, this is without the lens, and this is with the lens. So it makes a big difference. It means that we can refocus the beam at the target. And it's also possible. So I remember a few years ago when we published our first results on the lens that I just showed you. Insightec was very skeptical because they thought it was not very versatile, because you can only correct the beam for one location. But sometimes you might wish to shift the beam around. And so our next step was to show that actually it's possible if you move together the lens and the transducer, the correction is still valid. The idea behind that is the so-called isoplanatic angle that is used by astronomists, where they typically, if there's a small star here that they would wish to look at, but that they cannot see because of the turbulence of the atmosphere, what they do is that they use a reference star, a very bright star, that would be a little bit blurred because of the atmosphere. And then they adapt the telescope in order to see the star as a point. So they correct the aberration from the atmosphere, and then they look off and they see the tiny star. And we have the same here. We compensate the brain, the skull here, so we have the lens. We know how to target there, but if we wish to go close to this target, you see that the intercepting beam is very similar, so the correction should still be valid. So again, we validated it on human skulls. So basically, you see, this is without correction, what we measure through the skull. So you see, this is one skull in red, another one in green, another one in blue. And with the correction, we can refocus at the right place. This is at the central location. And then we steer the beam, so you see that correction is not good here, but we are still able to focus at the right location. And with the same lens, we can steer the beam off and off. And mostly, well, you see all the way to 10 millimeters away, it's still valid. We did the same also in the other direction here. We move closer to the brain. Again, this is the correction without the lens, with the lens. And once we move up and up, we see that here the focusing is not as good, but is still corrected by the lens. So it means that once the lens has been calculated for one location, you can still move your focus 1 centimeter away in each direction. So there's clearly a growing interest in using the lens. Other groups work on numerical models to test the efficiency of the lens, some 3D printing skulls, and we tested it on human skulls. And so I would say stay tuned. We are now, so our idea is to develop neuronavigated device for focused ultrasound. We integrate the lens here in front of the transducer, so the system is now ready in the lab. And our next move is to create a startup company called Sonomind. Our PhD student got a prize from the French government. So he's paid for 1 year, not to work on academic research, but to create the company together with us. You see, by the way, that this lens here is a little bit, it seems complicated with stripes, and the reason is that actually what we did is we can use the lens to compensate from the skull, but we can also use it to create bifocal focusing. So in that case, this is on purpose, and you would choose the exact location of each of the foci, and the advantage is that actually it's not only one transducer here inside. We have 16 elements, so it means that we can either focus the beam on this location, or together, or with, let's say, 10 millisecond difference between the two. So it opens avenues in terms of understanding the brain connectivity and modulating brain activity. So this is currently under development, but the first prototype has been approved by the ANSM, which is the French FDA, for human use. So now we can use this one. We are eager to start using it on humans. I would like to thank the Focused Ultrasound Foundation for funding this research, as well as the ANR and the Fondation Bettencourt-Schuller, and thank, of course, all the members of the lab. It's a big lab. Officially, it's only one team. So we are one team composed of 8 researchers and a lot of students, PhD students, and postdocs. Thank you for attention. I'm ready for questions.
Thank you for the beautiful talk. Very exciting stuff going on there. So, questions? Yeah.
Thank you. Very nice talk. I have a question that applies both for your monkey data and for your tremor data with patient. When you stimulate the frontal eye fields, you see an after effect of about 20 or 25 minutes, right? And you find the same when you do the tremor with your patients. So the question is, this is due to sort of plastic effect in neural terms, or is it because heating?
There is no heating.
No, in the monkey, you don't have heating either?
In monkeys, very few. The thing is, there is the water balloon that also decreasing the temperature while we are sonicating. So typically, the thermal rise was a little bit less than 2 degrees. So that there was some. The skull the temperature was increasing a little bit on monkeys. but not more than 2 degrees. And on patients, at least we could measure temperature with MR, so which is what we did, because I mean, it was already planned for the second stage of the treatment. The thermal rise on average, well, it was mostly noisy. It was less than 0.5 degrees, but on average, but you could see fluctuate a lot. It's not below the 1 degree that we would be able to measure with MR. And the reason why those two in time are similar, the duration of the effect is similar, because in both cases, the duration of the stimulation with ultrasound was basically similar, 20 seconds for the monkeys, 15 seconds for humans. I would expect that if we sonicate longer than 15 seconds for humans, we should be able to pronounce the effects. For sure, this is extremely preliminary work. I was very happy to be able to actually check that the maximum power that we use to design or prototype actually corresponds to the lowest power we used with the Insight X. So at least we know it's supposed to be efficient. But we need to test duration of the pulses, also the pulse repetition frequency. There are many things to optimize now. And it is too expensive to do it with the Insight X machine, because you need to pay for MR time. It's also not easy to use. So it's just paving the way. It's reassuring for us. But I think that neo-navigated devices are much better now to further investigate neuromodulation effects.
Thank you.
Thank you. Thanks a lot. I think you also nicely showed again another example of state-dependent effects, right? Even on the single neuron basis, those activated by a task are suppressed, those inactive during the task activated by the same protocol. But do you think that despite the maybe large impact of state, brain state, are there certain rules for constructing stimulation protocols, choosing the parameters to get bidirectional effects, inhibitory and excitatory? And I know, I mean, at the moment, a lot of labs are trying sometimes theory-based, sometimes trial and error, but like to find protocols that can do suppression or facilitation. Could you speculate on what rules might be found? Like how do you need to compose stimulation protocols to get one or the other effect, on average at least?
Exactly. Well, that's what I would like to investigate more. And I have some clues on those patients, for example, on the essential tremor. To be honest, we tested 2 sets of ultrasound pulsing, different sequences. I was kind of hoping that some would be inhibitory, some would be excitatory. It's very difficult now to analyze the results that we had. And the only thing I can say is that it takes time to build this effect. So for some patients, for example, and it's too bad, we sonicated, same way as for those 2 patients. We did not see an effect, and we switched to treatment. And actually, what we noticed is by looking back to the data that we had, is just that when we assessed the tremor right after the sonication, we did not see an effect. But fortunately, we also assessed 5 minutes later, and we saw a drop. So it means that it was coming, but it's not immediate effects. It takes time. It takes time, and I think that it's also due to the state of the brain. Patients were different, one from each other. And so those are things we need to investigate more, but we need to be able to spend time with the patients. With the rush of a clinical treatment, it was not optimum. But there are many things we need to do. First, take time, because it takes time to build, and then test different sequences, but not in a row as we were doing, because we tested them, but there was only 3 to 4 minutes between the stimulation. We should test one stimulation, wait for, I don't know, maybe half an hour, and then test different stimulation to be able to see the differences. There are so many things to test now.
Thank you.
And maybe one comment also, I'm not saying that we need to compensate the brain aberration because we are starting the company, but it's the opposite. We are starting the company because we believe this is what needs to be done now to move the field forward, to be able to ensure that we will concentrate the ultrasound at the location where we wish to stimulate. And in the past, I was surprised to see, for many years, I thought that using low-frequency ultrasound was enough, and that without correction, it was possible to do so. But when we do this retrospective analysis and look at the field distribution that was actually, we believe, was obtained on those human volunteers, now we understand why it was so disappointed.
I understand that the inside technology is much better deep in the brain. It has a problem with close to surface areas. So is your lens then able to target also closer to the surface area?
No, well, one could say that the lens, it depends on the resolution of the 3D printer, but mostly this lens is equivalent to something like 50,000 transducers, because we can create any shape we want. But by essence, it's not very different from the inside tech. So both systems would suffer the same limitations. The only thing is that the treatment envelope limited by inside tech at the center of the brain is only due to thermal safety. So it's just that if you move closer to the bone, then, as I showed, you know, if you look at without correction, okay. Here it is. See on this one, if you look at the skin, so you see here that the ratio between the pressure amplitude on the skin and at the center, if you move your beam further closer to the skull, you will end up having a pressure higher on the skin than in the brain. So if you're doing thermal therapy, it's traumatic because you're going to burn the scalp. If you are doing mechanical therapy, so BBB opening or neuromodulation, this might not be a problem. So with non-thermal therapies, you can use the lens or you can use the inside tech machine. And currently what they do, for example, for BBB opening, they go very close to the skull. This limitation is only for thermal ablation.
Just one question. Thermal ablation, the idea is that mechanical energy is transmitted to thermal energy and destroys the tissue. Mm-hmm. But then what would be the mechanism that this mechanical energy affects in the low intensity cells and cause the actual potentials or whatever to do? Because it's not the same thing. Mm-hmm. The fact that you have the same effect for tremor doesn't actually prove this, because the effect you have after your treatment is because the tissue is destroyed. It doesn't work. But when you do this low intensity, it's probably an inhibition, because that's the only way you get suppression of tremor. There is no other, that's the only. So that's a tricky thing, how this mechanical energy changed the activity of the cell. Why would this happen?
So there are many explanations possible. The one I like is flexoelectricity. So if you imagine one neuron, and when you apply ultrasound, and you concentrate ultrasound, you have what we call the radiation pressure. And we measured it on MR. Typically, the displacement of the tissues is a few microns, up to 4 microns. And so if you bend the neurons and the axons, then you're going to trigger action potential. And then once action potential are gone, then you're gone with neuromodulation. So I believe this explains why with pure mechanical waves, you can trigger action potentials. And then, depending if this is on excitatory neurons or inhibitory neurons, you might excite or inhibit a region of the brain. So you have somehow to make sure that your ultrasound parameters excite more inhibitory neurons, for example, to induce an effect. And you see this is a purely non-thermal effect. It's also possible to use thermal effects, because this will also change the rate of firing in tissues. Those are two completely different approaches. For safety reasons, in our lab, we prefer to concentrate on mechanical effects. And the prototype that I showed you, for example, the thermal rise is always less than 1.5 degrees everywhere into the head. It's just that I think it would be much easier to translate to humans that way.
So I guess even with the lens, if your transducer radius is limited, you will have a certain prolonged focal shape, right? So do you think that we're close to be able to use also cross-beam approaches using like 2 transducers, maybe even with 2 lenses, to precisely target a region at the point where the 2 beams cross, then with a much smaller diameter, only with the beam width, not length?
Our transducer is pretty big, so it's already tightly focused. So we can use the lens to focus closer or more or less to the transducer. But I would believe we would more use the lens to enlarge the focus rather than to make it more precise, because it's already maybe more precise than needed. I know it's possible to use intercepting beams,